नमस्कार, आभार, अभिनंदन। मैं आप सभी का इस्तकबाल करता हूं। गुड इवनिंग वन एंड ऑल। ओके, कैसे हो? हाउ आर यू पीपल? तमन्ना, स्मरणिका, यस। सो लेट अस स्टार्ट द थिंग. What we have done up till now only with respect to microsporogenesis, how pollen grains are produced. That is the recap of what we have done up till now. Now the question is, sir, you have not discussed that question, what most of you have done it wrong or incorrect. The question was very simple. It was with reference to polyspory, more than one spore, also called as polyspermy. Who does that? Usually monospermy. At a time, single spore pollinates. Polyspermy is done only in Cuscuta, Amar bale or Akash bale. It is total stem parasite, Cuscuta. Right? Now, coming to the goals and agenda for the day. So, when we talk about agenda, it is mentioned, has come onto your screen. We will be talking about counterpart of microsporogenesis, that is megasporogenesis, types of ovule, structure of ovule, and inside it, the most important component of the structure, that is embryo sac. That is what we will do it. Right? Hemant, fantastic. If question is asked, tapetum is diploid or polyploid? Tapetum is always polyploid. And I'm sorry, Nikit, क्या हो रहा है Nabil? Please concentrate in the class. Personal thing you can do anytime. Right? It's a request. Because others get distracted, if not you. Now, coming to megasporogenesis, structure of ovule, type of ovule, and structure of embryo sac. Sir, from where this business starts? Let us talk about that. When we say, you might have seen in a flower, the structure comes is this simple central structure you might have seen. On this is ovary. This is ovule, and when I say this is ovary, this is ovule, this is style, this is stigma. Perfect answer, Nisarga. Perfect. Tapetum is polyploid due to free nuclear division. Now, if you see, I will be talking to this complete structure today. That is what you call it as pistil or carpel. And in this, the most prominent structure that we are talking is ovule and ovary. Ovary, when ripens, it turns into fruit, and ovule, when it ripens, it turns into seed. Right? Simple business, direct answer. Now the question is, what does this all structure? Typical carpel contains stigma, style, ovule and ovary. Right? From here, let us move how the structure of ovule looks like. And when I say how the structure of ovule looks like, structure of ovule is an oval structure, and when I say it's an oval structure, I am just making it thick, otherwise it is a single layered. On the single layered, the most important thing you should always remember is these are the two integuments. And when these two integuments this is something that you have to understand it. Here this is the structure. This central core structure is called as embryo sac. These are the two integuments. This complete structure, what you can see which is coming towards this peripheral area, is called as funiculus. The structure or the point at which funiculus is attached, it is called as hilum. And when this hilum is cut or when funiculus gets detached, this point, the detachment is called as raphe. And here you see this opening is called as micropyle. Opposite of micropyle, this area is called as chalaza. Now, if you see this structure, here I have made it micropyle is close to funiculus, and technically if you see, if I make this diagram like this, yes sir, this is micropyle, this is chalaza. Opposite, when chalaza is opposite to micropyle or micropyle is toward the south pole, south end, this is called as anatropous ovule, and most of the books explain this anatropous, also called as atropous. Why sir? Because 82% of the angiospermic families, 82% of the angiospermic families exhibit anatropous ovule. I repeat, this is the structure of ovule. Sir, where it is present? As I told you, it is present inside the ovary, which is the distal part of the carpel. If you see this carpel, this is the distal part. Stigma, style, ovule. This is the stigma only on which pollen grains come, pollen tube elongates and reaches to ovule. This is the structure of ovule, which is a globular structure, which has a egg-like structure into the center, that is called as embryo sac. Embryo sac is covered or surrounded by two-layered integument. Sir, it is always two. 95% times it is two integuments. That is why it is called as bitegmic. That is why it is called as bitegmic, because two are there. Outer integument, inner integument, so it is called as bitegmic. Sometimes only single integument is there, that is called as unitegmic. Sometimes more than two are there, that is three integuments are there, that is called as tritegmic. That is rarest. Yes, there are the ovules in which there are no integuments, that is called as ategmic, right? Yes, further integuments develop into seed coats, and there are the plants in which integument becomes edible. Integument also becomes edible, right? Like myristica. In that it is edible integument, that is called as third integument, that is aerial in Myristica. You should always remember. And yes, as you said, Suraj, integument develops into yes, that is testa, and yes, Nisarga gave a certificate to it. Fantastic. It is testa and tagmen. You people are smart people here. I am so happy about it. And if you are very comfortable in this chapter, do remember you will get 5 to 7 questions in your NEET. 5 to 7 questions. I am very clear. Right? Now, this is the content. Funiculus is the very elongated strap-like structure which is connected by the hilum. If hilum is detached or funiculus is detached, it leaves a mark called as raphe. Opposite end of micropyle is called as chalaza. Micropyle is the opening through which pollen tube or water enters. I will slowly, when I tell you in the subsequent classes about fertilization, what is the significance of it. From chalaza, it is embryo sac, central structure which has egg apparatus and all. When I will give you the structure of embryo sac today, I will go ahead, I will tell you. Then, more importantly, I told you this is about anatropous ovule, right? Now, from here, the most important thing that you should always remember, function of raphe is unknown, is Marnica. It is just an attachment which leaves a mark. That's it. It has nothing to do with what is function. When a wound is there, a mark is left. That mark is called as raphe. Right? Now the question is, as your question was, I think somebody's question was whether it is attached to placenta is attached. Can be, placenta can be attached, right? Now from here, let us move to types of ovule, right? So when I said types of ovule, do remember unitegmic, ategmic, bitegmic. I have not given you the example. Bitegmic is very common. Unitegmic, ategmic, tritegmic. You keep looking into the books, and more importantly, bitegmic is very common. Unitegmic is found in Cuscuta. Again, unitegmic is found in orchids. Ategmic is found in Oncorona, and tritegmic is found in Asphodelus. Please, is it the same as on the gram seeds, the scar? Yes, but Anshuman, that scar leads to a whole sort of thing that is exactly the same, perfectly what you meant to say, Anshuman, because it's on the vertical suture in all leguminous seeds, not only on the gram seeds. Yes, on the leguminous seeds, Anshuman, perfectly. That scar is only raphe, right? Even you can see this raphe, a line-like structure in the seeds of what you call it as an oil seed, castor. In a castor also you can see that, right? Now moving to different types of ovules, that is important. I will just write the names first. Orthotropous, next anatropous, hemitropous, campylotropous, amphitropous, and sarcinotropous. I am happy people are doing fantastic job by making my job much easy. Yeah, I wrote Nisarga. Okay. In orthotropous, hilum is absent. Yes. Yes. Correct. Raphe is a mark, Rahul Shinde. Raphe is a mark. It's very simple, try to understand. Raphe is not an issue at all. In an anatropous ovule, when you see here, it has taken a turn. Usually this is integument, but integument takes a turn here. This point is raphe. This point is raphe, a bridge between funiculus and the base of an ovule. Otherwise, I'll explain. I think orthotropous, I think Nisarga told, hilum is absent. Yes. What is orthotropous? I'll go one by one to explain it. I'll just make one for you. Remaining you have to imagine, okay? If you see this structure, ortho means this is micropyle, this is chalaza. Here you see chalaza is opposite. So in orthotropous, the most important thing that you should always remember is micropyle and chalaza, they are in this direction. It is straight, ortho means straight, and orthotropous ovule is found in, as yes, she has answered me perfectly, Nisarga, polygonum. And that is the reason whenever we write, we always say polygonum type of ovule is. Now in your NEET exams, they don't give orthotropous, they give polygonum type of. Polygonum type of ovule is a characteristic. There they give the name, orthotropous, anatropous, right? And yes, micropyle and chalaza are opposite. Micropyle is towards the upside, chalaza is, see you can remember like this, chalaja in Hindi, chalaja, or you can say chal aja, come, you can take both ways, right? Chal aja, chalaja, that's a unique thing, try to understand it. So it's up to you. Chalaja, micropyle told chalaja, he went, but what he meant, chal aja, I want you. The time he understood it, in between other people occupied. That is what your competition is. We always tell you chal aja, you say, sir, you told chal aja, I went. So somebody else takes the seats between you, right? So do remember it. This is polygonum, that is orthotropous. Anatropous, I told you, that is inverted. This is also called as inverted ovule. Hemi means 90. Hemi means 90. This is the position. Micropyle is this. If you see, this is 90 degrees. Funiculus and micropyle. Most important. When I say this, body of ovule is twisted halfway. Body of ovule is twisted halfway. That is a unique thing. Is twisted halfway. But the degree of curvature is intermediate between anatropous and orthotropous. So neither this nor this. It is somewhere like this. That's why I told you. And this hemitropous is very common in ranunculaceae, most importantly. Most commonly, it is found in ranunculaceae. Do remember. Nisarga, P. Paplinaceae, correct. I read it something else, sorry. Cucurbitaceae, Solanaceae, Peplinaceae, all. I will write the conventional name of it. That is Leguminosae, pea family, Mutter family. Yeah, very good, Juhan. Fantastic, Juhan. I like it. Primula, fantastic example. Fantastic example. So pollen tube enter through integument in Hemipterus. No. I'll come back to it, Suraj. Have patience. I'll answer all your questions, man. Okay. Let us go to this. If I answer everything now, in fertilization I have nothing to teach. Right? धीरे धीरे खाओ, आराम से हजम होगा. एकदम से खाओगे, ill digestion, you know, food poisoning. Okay. Slowly, slowly. Okay. Breakfast be at the breakfast time, lunch at the lunch time, dinner at the dinner time. If you mix, it will become Megasporogenesis. Right? Disaster. So what is Megasporogenesis? Taste of nothing, flavor of everything. Please? Okay. Good. Coming to Campylotropous. Name itself says Campylo. Campylo is in which entire body becomes like a curve. Campylo, in which body of ovule becomes like a curve. If you understand this diagram, if you can see my hand, that is Campylo. Campylo. So that micropyle and chalaza lie either side of the funiculus. Something very interesting. If I say this is micropyle, almost they lie very close to each other, curved shape structure, right? When I say this, it's curved shape structure, it is Campylotropous. And do remember, but embryo sac remains straight. Try to understand here this. Embryo sac is not curved. Only the body of an ovule is curved. This body is curved. And when I say this body is curved, do understand one simple thing. This remains straight in case of Campylotropous. And example of Campylotropous is your mustard family. Caparis, Caparidaceae. Caparis, Caparidaceae. Now they have started asking the families. And mustard comes to Brassicaceae. Right? Coming to next, that is Amphitropous. Amphitropous. And usually people get confused between amphi and Campylo. In this, amphi and hemi look same. No. In this amphi, entire business is of embryo sac. This embryo sac becomes horseshoe shape. Embryo sac becomes horseshoe shape. This is something very important. Embryo sac. Body of ovule shows prominent curvature. It does not have curved and all. Has prominent curvature. And when I say it has like this, body is like this. Curvature, not orthotropous, not anatropous. Anatropous is this. Orthotropous is this, curved structure. But in this curved ovule, here is your embryo sac. Try to understand this, right? This is most important thing. And when I say this, and this makes the, what do you call it as, embryo sac becomes horseshoe shape, and what do you call it as, example is Capsella bursa posteris. Capsella, Capsella, Capsella bursa posteris. also called as shepherd's purse. Again, my dears and nears, this is the member of Brassicaceae. Family is the same. Ismornica, yeah, it is also found in few conifers also. Not in all, few conifers. Very good. Coming to something very interesting, that is Sarcino. And if you see this Sarcino word, I have used when I have taught you gymnosperms. Sarcino. So what happens here? Entire ovule is like an orthotropous, is straight, but funiculus makes a complete 360 degree. Funiculus makes complete 360 degree. Means it covers the complete embryo, complete ovary. If I assume this to be my ovule, this is the funiculus, this funiculus curves completely. When I say it covers completely, this is called as sarcino. It is like a mother who is holding the baby. It's just, I hope you have seen how a mother monkey holds the baby monkey under security. This is what exactly is Sarcinotropous. And when I say Sarcinotropous, classical example is cactus. All the xerophytic plants. Cactus is the classical example, Opuntia, cactus. Right? Please, you understood this? So what we have done, orthotropous, anatropous, hemitropous, campylotropous, amphitropous, sarcinotropous. Examples, ortho straight, ana inverted, hemi 90 degrees, campylo curved, amphi horseshoe, sarcino completely covered. 5 words, that's it. Ortho straight, ana inverted, campylo curved, amphi horseshoe, hemi 90, sarcino completely covered. Understood? Contest time. And yesterday's that question, whosoever has answered is, question was fertilizin, and the fertilizin is chemically, it is glycoprotein, and Tamanna Sharma, she has secured 1 atom session. Congratulations Tamanna, I am happy people are competing. And now it is today's contest question, mine, 30 seconds, and question is, the edible part of lychee is, that's it, edible part of lychee fruit is, sir what is lychee? Lychee chinensis, a type of fruit, edible part of lychee is, please give me the answers. I think somebody answered earlier. Okay, 30 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Full stop. Good. I am happy at least so many people are answering. I am so happy about it. Right? Now, let me come to a structure, or to tell you the structure of embryo sac. Okay? Because once I tell you the structure of embryo sac, how megaspore happens and all becomes secondary. Only from here you should remember the ovule, as well as you should always remember second thing, that is embryo sac. So let me tell you about the structure of an embryo sac. And again, as I told you, it is a globose structure, and in this globose structure This is the central vacuole, and as this is 3-celled stage, here also 3 cells, here also 2 cells, right? Simple. If you see here, the total structure here are 8 cells, right? This is the structure. Sir, what way? Okay. This is micropyle. This region is called as chal aja, nahi to chala ja. Okay. Now, when I say this micropyle or chalaja, the most important thing you should always remember is, here 3 cells are there. This central cell is called as egg, and surrounding the egg or the bodyguard of egg are called as synergid. You know, it is like 3rd division. Sir, somebody asked me, I asked one student, hey, in SSLC, what grade you got? He said, sir, 1st division with 2 bodyguards. I said, something interesting, I never realized it. He said, sir, I got 1st division with 2 bodyguards. I said, gentlemen, tell properly. Arey sir, 1st division with 2 bodyguards. I said, wow, perfect way to explain 3rd division. See, 3rd division you say it becomes problem. But we say 1st division with 2 bodyguards, it's a pride. It is like dirty shoes covered in a velvet cloth, right? So, I am also 1st division with 2 bodyguards, teaching to the people who are only 1st division, right? Tum tum talam talam, right? Bindas. So, egg surrounded by synergids. Opposite to micropyle is chalaja. Chalaja has 3 cells and all the 3 are called as antipodal. Time to time various concepts have come, so many come. They say ki they are the vegetative cell of embryo sac. Some say ki yes, when it is vegetative cell they provide nutrition. Suraj, they are not the sister cell by default, okay? Congo sir, yeah. What is that Congo sir? Congo is a country. I am not from Congo. Okay, I am Indian. Okay. As I told, antipodal, they are the vegetative cell, and during the time of fertilization and after fertilization, they provide nutrition. They are motherly in nature, right? In the center, as I told you, this is a central vacuole. Central vacuole has 2 polar bodies. Here we will not write polar bodies, otherwise these people who do oogenesis, they will kick me. Oh, Congo for congrats, but don't make so much shortcuts, okay? Congrats for what? Okay, for giving you the question or answer, okay? Polar nuclei, and when I say this, these polar nuclei in a due course of journey becomes polar nucleus. Why sir? Because these 2 polar nuclei fuse to become 1, and when it becomes 1, it becomes polar nucleus. So if you see here, this structure technically is called as seven-celled 8-nucleate structure. 7-celled 8-nucleate structure. Why sir 8-nucleate? Egg 1, synergid 2, polar nucleus 2, antipodal 3, and if you count 3 plus 2 plus 2 plus 1, it becomes 8 nucleate. Sir, how many cells are there? Egg is 1, synergids are 2, polar nucleus is 1, and they are 3. 3 plus 1 plus 2 plus 1, 6 plus 1, it is 7-celled 8 nucleate stage. 7-celled 8 nucleate stage is an embryo sac. Please try to understand it. This embryo sac, whenever we explain, we always explain this is as a monosporic or monospermic. This is what the polygonum type we explain. Why, sir? Because micropyle is up, down is chalaja. We are talking about orthotropous, or what you call it as polygonum type. Right? This is about your embryo sac. Now the question comes, sir, how this entire structure develops? And in the future, I will tell you, this polar nucleus only gives rise to endosperm, tendril coconut what you drink. In fertilization stage, I will tell you, I will explain you. Now coming to the last part of it, that is megasporogenesis. How this megaspore comes? When I say this, how this megaspore comes, it is something very interesting here. It is formation of megaspore from the megaspore mother cell. Megaspore mother cell giving rise to megaspore is megasporogenesis. This is 2n, this is n. Simple. When a diploid structure becomes an haploid, it is called as megasporogenesis. Same exactly like microsporogenesis. Only difference is in microsporogenesis you have talked about tetrad and isobilateral structures. Here there is no successive type, no simultaneous type, no monocot type, no dicot type. Here it is straight, simple business. Right? People are getting it or not? Yes, my dear friends, मेरा बच्चा लोगा समझ आ रहा है? एकदम से आप लोग रुक गए. मानसी, हेमंत, सूरज, तमन्ना, गणेश, स्मरणिका, Nisarga, so many people. Are you able to follow me? Otherwise it looks like I am only following myself. Now, where it starts? It starts inside the developing. This journey of entire business starts from the most importantly, try to understand, very early when nucellus is not completely surrounded by the integument. When I say nucellus is not surrounded by the integument from the beginning, that time only. Yes, Rahul, secondary nucleus and polar nuclei both are same. Yes. And when I say when nucellus is not completely surrounded by the integument, there only it happens in the developing ovule. Sorry, I could not complete it. Developing ovule of an angiosperm. One of the cells of the nucellus, this is the nucellus, so many cells are there. If you assume like this, one of the cell starts proliferating anticlinally or periclinally, and this becomes archisporium. This archisporium slowly differentiates, as I told you, into a megaspore. Do remember, this archisporium proliferates into megaspore mother cell, and this megaspore mother cell gives rise to 4 megaspores. 4 megaspores, and they are arranged like this. And when I say they are arranged like this, the most importantly, top 3 degenerate. Top 3 degenerate. Only one becomes functional. Lowermost megaspore becomes functional. And do remember, which is situated towards chalaja. There only I always tell the meaning. Everybody thought chalaja, so all got thak. Chal aja. Then he realized, oh, my chance came. And that is the reason the functional megaspore at the chalazal end becomes functional, and this is the chalazal one which has the nucleus. As I told you, this megaspore undergoes 4 mitotic divisions. When I say 4 mitotic divisions, it gives rise to 8 nucleus, and this is the 8 nucleus which I told you, 7-celled 8 nucleus gives rise to embryo sac, which I have already explained you, right? Do remember here, I have to tell you, in case of cypress, that is cypressy members, all 4 become functional. All 4 becomes functional. Even it is 99% times it is correct that chalazal one only becomes functional. There are the cases in which 1 towards micropyle becomes functional, 3 towards chalaja becomes non-functional. 1 towards micropyle becomes functional, whereas 3 towards chalaja becomes non-functional. That is very rare. You don't have to bother about. But in cypressy members, do remember the most important thing, all the 4 becomes functional. All the 4 becomes functional, right? So, what all things we have done up till now? In a nutshell, let me tell you. We have talked about, I have done reverse engineering if you see. I have explained you ovule first. When I say I have explained you structure of ovule in detail, from there I moved to something called as types of ovule. From types of ovule I have explained you embryo sac. From embryo sac I have explained you how this entire embryo sac formation happened. Importance of meiosis in MMC, it is responsible for megaspore formation, and this is the megaspore that is giving rise to embryo sac, female gametophyte. If meiosis would have not happened, female gametophyte would have not happened, female gametophyte would have not happened, we would have never got endosperm. I will come back to it, Juhan, filiform apparatus and all I have not told you now. When I say I have not told you obturator, function of obturator, filiform apparatus, hypostat, all that will come in fertilization. Please try to understand it, right? One thing I will tell you before I close, nucellus, which is diploid structure in entire embryo, entire surrounding embryo sac, this nucellus is either multi-layered. When it is multi-layered, it is called as crassinucellate, very heavy, too much eaten. When it is not eaten, single layered, it is called as tenvinucellate. And that is the reason when you see a big embryo, a thick embryo, do remember it is crassinucellate, and when you see pulses very dry, you call it as tenvinucellate, right? So, all my dears, all my friends, all my passport users, start solving the questions now, immediately. So all of you, take care of your good health. May God bless you all. See you soon. Till then, Shabba Khair, Shubh Ratri, bye bye.